Nice introduction. So I really think that I was lucky in the last 10 years to work with great people, great mentors, Dr. Yang, Dr. Bell, and all of those people that they support me. So, and also great uh, colleagues that I had. So this is, I think, this is very good luck for me that I had. So, and thank you so much, Peter, for all of those nice things. So I started my lab about 4 months ago, and my lab is working on synthetic biology for translational research. So first, I'm going to talk a little bit about how I heard about synthetic biology, and then also talk about a little bit about what I'm doing, I did during my postdoc, and also what I'm doing here. So it was, I think it was 4 years ago. So let me. So every year there is a meeting. Its name is Lindau Nobel Laureate Meeting. So all of the Nobel Prize who are alive, they get together and they talk about the future direction of science. They talk about what is the challenge for humanity and also what we should pay attention and how it would be the future of science. Also, it is very important meeting, and some of the thing that is very famous, it first came from this meeting. For example, the agreement about the climate change or non-proliferation of nuclear weapons, first it was discussed in this meeting. And it's very interesting that this meeting, there is also a website that all of the student, PhD, master, and also postdoc, they can go to the website and register for it. So they gave you 5,000 euro and they pay for your travel, they cover you the ticket, they cover you the price for the ticket, and you go there and spend 1 week with the Nobel laureate, which is great opportunity. And I really encourage you to do that. And if you don't have time for making the application, you can go and watch the videos. Every year after the meeting, they upload all of the movies. So with help of CIHR and also Dr. Yang, I was able to apply for this meeting. I was honored to be there. And it was 2000, I think 18 or 19 before the pandemic. And it was three key things that they consider as the most important thing for the humanity and the challenge for the humanity that we should pay attention to. One of them is the climate change. One of them is the emerging infectious disease and antibiotic resistant bacteria. And it was before the pandemic. And it was very interesting that this year I watched the video and they said that we said that it's important and we should pay attention to it. Pandemic is very important. And now we are saying that pandemic is very important and we are not still ready for the next pandemic. So we didn't get listened because of, there are many reasons that they discuss about it. And also we had a course there. It was, I'm not saying that is the best description of being successful, but it was something that I liked it. So I wanted to share with you, and maybe some of you heard about it, that is ikigai. So ikigai is a Japanese word that it's the concept of for a reason for being. And it says that to be one way to be a successful, definitely there are many ways that we can be successful, but one of them is that you find something that you really love, something that you can get money for it, something that it address some challenge, it address some need for the world, and also something that you are good at it. If you can find something that in yourself that it has these 4 criteria, so you will be happy because you are good, you can get money for it, and also you are doing something useful. And it's something that every time, every 6-7 months, when I start a project, I ask this question from myself again, that am I doing something that I'm good? Am I doing something that addressing something? And am I enjoying it or not? So it's very important to find your ikigai. And during the meeting, also, I had a great chance to meet some of the Nobel laureates. One of them was Richard Roberts. And it was very interesting for me. He was saying that he didn't find his ikigai. He got Nobel Prize in alternative splicing, not alternative, on splicing and finding intron, if I'm right. And he said that he is enjoying something else. And even after he received the Nobel, he changed his field. And now he's working with NEB. And he believes that, he was saying that it's very interesting that for almost every question that we have, there are 4 billion years experience in nature. And nature has a lot of resource that we can go and find those resources and somehow repurpose it for some of the questions that we have. And he is developing novel synthetic biology approach now for NEB, and I found it is interesting. And also in the meeting, I heard about using bacteria and virus to target the cancer. So I thought it's interesting. And also I know about myself that I enjoy basic biology, but the most important part for me, it's not basic biology, it's developing tools. So I know that I'm interested to get familiar about new tools to address something, some question, some biological question. So what we are doing now, my lab is focusing on synthetic biology and synthetic biology for developing novel technology for translational research. And synthetic biology is a multidisciplinary area of research in which you use different resources from nature to address some industrial, medicinal, or environmental application. And today I'm going to talk a little bit about each of them, but mostly about oncolytic virus. So now we are trying to get funding for some of them. We were lucky, we received some support, and we are trying to get about the rest. But first, I'm going to talk about oncolytic virus. So oncolytic virus, we heard a lot of bad things about them, but today I'm going to talk in defense of the viruses. Viruses can be used also for good things. And what is the idea? First, I put two examples, and I'm going to tell you what is the basic idea, and then explain that what would be the future and what we are interested. So here in this example, you see that there is a patient, Dr. Bartler, he injected the virus in a specific location, and you see a good response in the tumor, metastatic tumor, after 90 days. And in this experiment, they use Western Reserve, which is a vaccinia virus. I will a little bit explain about this virus. And in another example, you see that it was melanoma metastatic and it was injected, the virus, they injected in multiple locations, and then they see a response. So what's happening? The story started 50 years ago. They saw that some patient who have cancer, after getting infected by virus, they get flu, they get a little bit recovered from cancer, or they get a little bit better. And then they came with this idea that we can use virus to maybe treat the cancer. And what is the idea? The idea is that first, here, for example, if you just look at as an example, if you have a cold tumor, so when it's infected, you have somehow inflation, and then you have the recruitment of immune system to make that tumor from cold to hot. This is one of the things that can happen. Or you can remodel tumor microenvironment from being cold to make it, from being suppressive to make it more active. So you can use the oncolytic virus to remodel the tumor microenvironment. But the basic question is that why virus? They should like to grow in cancer cells. Why the virus, they are not growing in the normal cells? First, some of the virus, they grow in normal cells as well. But the virus that we are trying to use for cancers, somehow we try to engineer them to be more specific for cancers by putting some genes or removing some genes from them. And also, we should not forget that cancers in general, not all of them, they have a specific feature that it makes them attractive for virus. During cancer progression, usually we have inactivation of interferon pathway. So interferon pathway, which is important for response to viral infection, usually it's suppressed in cancers. And because this pathway is suppressed, so it's a good environment for virus to grow there. So when you inject the virus to the body, the virus, probably it goes to multiple tissues, but it's able to grow in cancer tissues because you have inactivation of interferon pathway, which is very critical for responding to the virus. So we use this feature to use viruses as oncolytic virus, to use a virus to treating cancer. And it's very important to know that we have many viruses. We have many viruses that it's been showed that we can use them as oncolytic virus. But currently, there are three main oncolytic viruses that we see some efficacy and some safety for treating the patient. And these three are VSV, which is RNA virus, is HSV, herpes simplex virus. It is a DNA virus. It is about 200 kb size, and it has the capacity coding about 25 kb. So you can put 25 kb gene in it. And the third one is vaccinia virus, which is also around 200, but the capacity, the coding capacity is higher. So vaccinia and HSV, they are DNA virus, and both of them, one of them is replicate vaccinia in cytoplasm, HSV replicates in nucleus, and VSV is RNA virus. It's replicating in cytoplasm, but all of them, they showed high safety. And so far, there is no integration of the virus genome from this oncolytic virus to the human genome. So it's pretty safe. But what is the general idea? The general idea is all of these viruses, because of the interferon pathway, they would like to grow in cancer cells. And based on our experience in the last 30 years for using these viruses, there is not that much off-target growing in normal tissue. And this is a paper that I didn't have time to read it carefully. It was published last week, and this is it was published in Nature. And what they did in this paper, there was glioblastoma patient. They inject HSV into the brain, and they look at what would be the survival of the patient. So, and they show that they can increase the survival average about 10 months. So this oncolytic virus that it's used here is just oncolytic virus without any cytokine or chemokine. So virus alone, definitely it can kill cancers. And also because it's killing the cancers, it's releasing antigen from immune system. And as I said, it also makes the tumor a little bit hot, and it activates also immune response. So because of all of those, you have a good response against the tumor. But we should remember that oncolytic virus is just like a vector. So just think about AAV, just think about adenovirus. So you have a virus that is killing, it's okay. If you want to get the most out of it, it's better to arm it with some cytokine. And when I started my postdoc, I saw a few papers that one of them, for example, they were trying to put one cytokine in HSV. There was another paper, they were trying to put another cytokine in another oncolytic virus. So we asked this question. We said, let's look at for 20 top cytokines that different people they are trying, and put it in vaccinia and HSV, and also VSV. So I didn't put the data for VSV here because now currently I'm rescuing the virus. So what's happening here? So first, let me explain this graph. So this is B16 model, this is C26 model, and each of these are representative of average of 8 mice. So for example, let's talk about PBS. So you have a PBS here, and when you have a PBS, I mean, you treated the tumor with PBS, with nothing. And after about 19 days, you see that it's getting more than 4,000. So this is endpoint. When it's yellow, it means that the mice, they're getting to the endpoint. And after that, we don't have any mice. So the blue here, it means that the tumor, they have a smaller. So here you see the scale for the size. So you have a PBS, you see what's happening. You have a just vaccinia as the vector alone without anything. And you see that it's working to some extent just by postponing the tumor growth for B16. What happens if we put interleukin 2, 7, 12? So, and what happens if we put it in HSV? What happens if we try other tumors? So what is the take-home message from this slide is each virus, it works differently based on what tumor you choose, and also based on, so for example, if you have interleukin 12 in vaccinia is working differently from HSV. Look at it here. If you have, one of the things is very interesting for me is interleukin 10. Interleukin 10 is kind of immune suppressive. And it's very interesting. When you put it in an oncolytic virus, it increased the efficiency of oncolytic virus. So because it makes sense, because it increased probably the persistency of the virus. The virus stays in tumor microenvironment for longer time, so it can kill more, which is consistent with a couple of papers, a publication in the past that they put suppressor of tumor microenvironment and they show that it's increasing the oncolytic virus efficiency. So this is a lot of data, and we can talk a lot about it. But my conclusion from this slide is you have, based on the virus, oncolytic virus you use, and based off the tumor environment, you need to arm your oncolytic virus with different cytokine or chemokine to get better response. For example, for vaccinia, what would you choose? I would choose interleukin 2, 12, and 18. So now I'm going to use interleukin 2, 12, 18, put it in oncolytic virus and treat patient, or first try some preclinical model. But the first thing you would say that 2, 12, 18, each of them is already a little bit toxic. If you put it in virus and put it in a patient, it's okay that is expressing locally in tumor microenvironment, but still there is a chance for cytokine storm. So you should be able somehow to control those toxicity. Then here, I ask the next question. So now we want to make a safety switch, not for only the virus, also for expressing those cytokine. And my plan was that, okay, I am going to publish some of this data for my postdoc, and with my permission of my mentors, I'm going to use the rest for different models. For example, also put pancreatic cancer, ovarian cancer, make it a bigger story and work on it in the next 5 years or 10 years during working in my lab. So now I'm going to talk about this part, but the rest is something that currently I'm doing in my lab with the students. So what was the next question? The next question is, we are going to express 3 top cytokine, interleukin 2, 12, 18, in vaccinia virus, but we need to develop safety switch. And this safety switch is something that we need to be able to control the virus replication, because oncolytic virus, I said that they are safe, but still sometimes they show off-target growth. So we need to be able to turn off the virus whenever we want, in case if they show toxicity. And also in your house, you don't have one switch for the light. You have multiple rooms, you have multiple lights. So the same thing for virus. You need to have multiple switch, because probably you would have multiple things that you need to be able to control it. So if you have a cytokine that is activating immune system or suppressing, or if you have a chemokine that is recruiting immune system, so for playing with the time and level of those cytokine and chemokine, you need to have a tool that you can control the expression and the time of those. So look at this ugly electrical car. So who wants to have it? But me and my wife, definitely we would love to have one of those Tesla car. So what happened about the electrical car, I would compare it with inducible system. We had inducible system, and I worked during my PhD here, and also I worked with them, but the problem is that I would compare them with those ugly car, that they have a leakage. Sometimes you see that it's working, but after a couple of weeks, the inducible system now is not working anymore. So our cells should be fresh. All of those problems, it can be the same for oncolytic virus as well. So we need to have a safety switch that has less leakage, better, it can be controlled in better way. And probably we want to have something that we can control it with FDA approved drug, because it is FDA approved drug, we can translate it to the clinic. And we work on a little bit AAV, a little bit HSV, and also vaccinia virus, but I'm going to focus on one of them, vaccinia, because it shows what I'm trying to show you, and you can think about the same way about other viruses. It's the same idea. So what is vaccinia virus? Vaccinia virus, I really like this virus because it's very fascinating. It has its own DNA polymerase. It has its own RNA polymerase. It has about 200 genes that it's interacting with different protein in hosts. And some of these genes are essential, some of these genes are not essential. So you can, if you knock out some of those genes, you can make this virus more specific for cancers. But another thing is that these genes are expressing in different level and different time. And as I said, it has its own transcription. So if I want to develop a system for controlling this virus, first I should know how this virus it works. So I should know when and to what level is getting expressed, and get the promoter to put it on something like luciferase and try to understand how it's working. And then we can, if we can establish it for luciferase, we can also try it for interleukin 212 or other things. So the first thing we did RNA-seq sequencing. We infect normal cells, cancer cells, and we tried to find some promoters that is working in both of them, because this will be used to specifically for engineering the virus to make it specific. But I want to make sure that I use a promoter that if I use this promoter, is probably is not going to work with cancer. So we chose a bunch of the promoters, and the promoters are 20 base pair. And we did RNA-seq to find the time that they are getting expressing and the level. So here I just you can show I showed the level, but also we look at the timing as well, and then we pick up those genes. The 20 base pair, we know that the printing for the RNA polymerase is 20 base pair. We pick those genes, we put 20 base pair, and we designed this very simple tool. So this is a vaccinia promoter. We put TET promoter, which is TET response element, and we put a TET expression with a constitutive promoter of vaccinia. So what's happening here? You have a TET repressor that it's binding to the element that is after of the promoter that I showed here. And we tried different promoters. So we have different genes that I showed you in previous slide, and we put a repressor element that binds to TET, and then without DOX, what we would like to see is that they are not getting expressed, because we don't want to have leakage. So you see some of them, they are getting leakage. And after DOX, we want to have to get the best expression. So you see some of them not getting expressed. You don't see induction very well, but two of them, it was working very well. So we made DOX inducible system for vaccinia, and we were able to control the expression. As you can see, 2 hours, 3 hours after adding the DOX, we can see the expression of our gene of interest, that here is GFP luciferase. And then we tried it in vivo in mice. You see that we showed that if it's off, we can turn it on. And if it's on, we can turn it off by removing DOX. Here we add DOX. So, but you would say that, Taha, you mentioned that you are going to make a system that is not like that ugly car, but it would be, you have some leakage here. That's correct. So we made a system, it's that ugly car. So, but can we find a better system? So let's go to the bacteria, let's look at the nature, let's look at other inducible systems. So we made an inducible system, it's a leakage, so it can be used for something that is not very toxic. But if it's toxic, we need a better system. We look at multiple systems, and one of those systems that bacteria is interested is QMAT response element. So this QMAT is being used as a food additive, and it has a sour taste. So when you get a candy, to some extent, they have this QMAT. So it's pretty safe, and it's approved by public health, and we have it in many many candies as a food additive. So how it works is very similar to DOX inducible system. So you have a response, QMAT response element, you have QMAT repressor, it binds, it doesn't let RNA polymerase work. And now we want to adapt this one, that's for bacteria. Now we want to adapt this for oncolytic virus vaccinia. So what we need to do, we need to try different combination of vaccinia promoter with different orientation and lengths with QMAT to see which one is working. So we did the same thing that we did with DOX, and as you can see here, one of the combination, it was working the best. You have expression, high expression, you didn't have leakage. And now we went for in vivo study. So first, you see that it's almost no leakage compared to the DOX. We tested in vivo and in vitro. And another interesting thing that I didn't put the data for DOX here, but it was interesting for QMAT that we were able to get different level of expression for QMAT when we use different amount. So you could get expression low amounts, medium, and high based on how much cumate or candy you give to the mice. And also, if you wait for 4 days, you can turn it off. So this system is working very well. So far, I showed you two systems. One of them was cumate, one of them was dogs, but I would like to have more. Maybe we should need to develop the third one, and then I tell you what would be next. Do you think three is enough, three system? So what is here? So after you make a new system, and this is the first time that it was tested in mice, you need to show that it doesn't have toxicity. So we did multiple study on mice to make sure that they are safe. If you feed the mice with high level of cumate for 50 days, and even after 50 days, we didn't see any toxicity, liver toxicity, and we checked about 10, 12 enzyme, and here you can see three main enzyme that is representative of liver toxicity. And then we said, okay, we have two, let's make the third system. So what is the third system? So I'm going to simplify it. So T7 is bacteriophage. So we use T7 RNA polymerase. We cut it into two, and then it fused to FKB and FRBP. So it's known that if you add rapamycin, what happens? It promotes the complementation of any protein that it binds to these two. So when you add rapamycin or rapalux, you have complete RNA polymerase that now it can transcribe your gene. So, and the good thing about rapamycin is that there are some rapalogs that is very similar to the structure with rapamycin, but also they have some anti-cancer activity. So you can use kind of combinational therapy. And again, when you have rapamycin, you can have expression of luciferase to different extent. And here we saw a little bit leakage, but we can turn on the system or turn it off. So far, I'm going to have just one more because now it's getting boring. So we are keep saying, okay, have another one, but I want to just say one more thing, and then I want to make sure that you are bored, and then go to the next slide. So now we have three system. Is three is enough, or you want to keep going? So I want to say that we can also combine these three and make it 19, 20. What does it mean? How can you make unlimited number? So this is just two examples of 12 systems that we made. So what you see here, we have, I showed you T7 inducible system. And then also after T7 inducible system, we put TET response element. So you have a system now. And if you remember, I said already that TET has a little bit leakage. So maybe this leakage is good. Maybe you can combine these two systems. And when you have none of them, you have two safety switch working at the same time. So you have very strong control of transcription of your gene, like what you see here. So to get express, you need T7, which is rapamycin, and then you have also, you need DOX to fully express. If you have none of them, it's a very tight control, you don't have expression. If you have DOX, as you remember, it was a little bit leakage. So you have 7 times expression compared to the background. If you have RAPA, you have more. If you have RAPA and DOX, you have fully activation. So what happened, just to make it simple, we made 3 systems, and then we just said, okay, we have 3 promoters that is inducible. Now we want to combine them. So when we combine them, now you have a new system. To be fully active, you need both compounds. So you have a new system that you can have 2 features. First, you can get different level of expression, nothing, very high, or medium, or low. And why it's important? Maybe your patient is adult, maybe your patient is old person, or maybe your patient is kid. Different patient, they have different level of activation of your transgene. The interleukin you are using for different patient is different. And so by using this new chimeric system, you can control the level a little bit more. It can give you different level based on what combination of use, and also it reduced the background. So to make the story short, we made multiple system, 3 system, and then we combined them, and then we made more. And now I'm going to show you some example how we can use this system to make some safe therapeutics for different for cancer treatment and also vaccine development. So we made inducible system for ducks, if you remember, and it was it has a little bit leakage. So, and this is actually what we need for vaccine development. When you have a vaccine development using, what is the example of vaccinovirus that being used as a vaccine is the one that we are using currently for monkeypox. So the monkeypox vaccine that we are using now in hospitals, some of them, they are coming from vaccinovirus and MVA. So in US Army is mostly from Tiantan, but also it's from MVA. So those are, so vaccinia can be used as a vaccine. So this vaccine, and also you can put some transgene in it to make a vaccine for other disease. But in manufacturing, you should be able to manufacture it easily. You cannot have a safe virus. In manufacturing, you need to be able to produce as much as virus possible. But when you inject it to the patient, it should be safe. And you should be able to have minimum replication. So in this example, you see that when you have a virus, when this virus in manufacturing, you can use ducks, and without ducks, it doesn't have replication, which shows here that compared to the one that is currently using for monkeypox, you see in immunocompromised mice, you see a lot of pox lesion, but for the other one, you don't see. And also when you look at the off-target growing is much safer. So now you have a virus that you can control the level of virus growing for manufacturing, and also it can be used for cancer therapy, because when you use this oncolytic virus, if the patient, you use this virus with dogs, and as soon as the patient show toxicity, you can remove dogs, so it shows some safety. So another example is fusogenic protein. So fusogenic protein, they are protein that they fuse cells together. And when the cells fuse together, usually it's followed by apoptosis and activation of immune system. So, and this protein is being tried in the past to be combined with oncolytic virus. But as you can guess, if the oncolytic virus, they show some off-target growing at the beginning, expressing this protein, it shows a lot of toxicity. So the idea we had here is at the beginning, you don't express your fusion protein. You just wait the virus to be established and start replicating, because when the virus is replicating in normal cells, they cannot replicate, and you wait for few days, and then you administer with ducks. Then the expression of those fusion protein is something that you see here. So at the beginning, you don't have fusion, but when you have an expression of those fusion protein with inducible system, then you have a fusion, and probably getting better response in targeting cancer. And here you can see that, for example, in these two tumors, you see without ducks treatment and with ducks treatment. It's very interesting. When you treat ducks with mice, you see in the tumor from A549 xenograft model, you see that it's the fusion, it can be clearly seen in the tumor samples, and we get a sample from multiple organs, and compared to when you continuously express it, we didn't see expression of those fusion protein in any normal tissues. And also when we look at the efficacy, you can see that compared to PBS or the virus that doesn't have ducks with fusion protein, you don't see any treatment. So what's happening, just to make this, to explain briefly. So we have a fusion protein. The fusion protein, the idea is that it's fusing the tumor, the cancer cells, because of the feature that this protein has, and it shows when fusion happened, it caused a lot of anti-cancer, but in the past that is tried in clinic, it was toxic. So we just put on inducible system, and now we were able to use this oncolytic virus for some preclinical model. Now let me get back to the original figure that I had. So I said, we did this study with many mice. We found interleukin 2, 12, 18. And we want to combine them. And I have two questions. My first question is that what level expression of interleukin 2, 12, 18 I should get to not see toxicity? So one of them is the level. And the second is, what is the time? Should I express it when I have a virus at the beginning, later? So what is the time? So now I have a tool that I can look at the expression and the time of cytokine and see which one can be show the best efficacy in treatment of the cancer. So we tried multiple systems for interleukin 12, 18, and 2, and finally we were able to make these systems. So all of them, we use QMate system. Why we use QMate? Because in the system that I developed, QMate was the best one to get dynamic range, and also it has lowest leakage. And we showed that we can control the expression with a QMate. So when you don't have a QMate, you don't have expression. When you add a QMate, you have an expression of interleukin 2, 12, 18. And this is just by ELISA. And what these three figures showing, this is, I'm going to talk about this one and a little bit about this. So here we have a vaccinia virus that is expressing interleukin 2, 12, 18, and they are under inducible system. We just, this graph shows that you can control the level of expression in vitro based on how much QMate you use. So this system, it shows that you have an inducible system for 3 different cytokines, and you can control the level. And now that you can control the level, first, is it really toxic? Do we need inducible system or not? When you have continuous expression of interleukin 2, 12, 18, yes, all of the mice, they die in less than 5 days. So we need inducible system. So let's put it under inducible system, but don't try the low dose. Let's go for high dose. And when you try the highest dose, again, you see that 80% of the mice, they die. So you have an inducible system. If you fully activate it, the mice die. Now, what is the level that we can try to have a safe range for treatment of into the patient? And also, we here it's 2 main experiments. I'm going to talk about this too, that what we look at as a toxicity. So the mice, before they die, the 2 main feature they showed is cytokine storm, which you can measure it by different enzyme, liver enzyme toxicity, such as, for example, alkaline phosphatase, and also the amount of water in the lung. So this is 2 main symptom that we see in the mice couple of days before they die. So we can just measure these factors to see the toxicity. Now we know that, so if we fully activate it, it's killing the mice. So we can try different level of QMate, 6,000, 2,000, 1,000 in their food to see which one is not killing the mice. And as you can see, in 1,000, is not killing the mice anymore. So we can consider it as a safe treatment. And here we look at, we'll use different amount, and we show that 1,000 is not toxic. But the question is that, are we seeing interleukin 2, 12, 18 when we use 1,000? And when we use ELISA, we saw that yes, for 1,000, we see good amount of interleukin 2, 12, 18, but definitely is compared to when we fully activate it. And this 1,000, when we look at water in lung or measure different enzyme activity, we didn't see toxicity. So now we find the concentration for interleukin 2, 12, 18 that is not toxic, but would be efficient in treating model or not. We tried multiple models. One of the models that you can see here is MC38 model, and we tried different time, and you can see that when you express interleukin 2, 12, 18 at the beginning for 5 days, you see about 40% cure. So I'm not to talk about other tumor microenvironment analyzing we did. Just this is, we developed a tool that we can control cytokine chemokine expression. And after that, we found what is the best dose, what is the best time to use it for treatment of a cancer. And now this is just the beginning of the story for different cancer. You have a breast cancer, you have a lung cancer, you have an ovarian cancer. In ovarian cancer, you see a lot of heterogeneity. In breast cancer, you see different, depends on what cancer you are seeing. Now you can go to different cancer model and look at that table that I showed at the beginning. Use the table again, or maybe repeat the experiment again in that cancer model to find what would be the best combination treatment. And also what I did here, I just used vaccinia. But for some of those cytokines, you can use vaccinia. For some of them, you can use HSV. For some of them, you can use VSV. The experiment can be also more complicated, not just using one virus. Now you can use different viruses. So now you can use this tool to hopefully efficiently remodel the tumor microenvironment. So I'm going to show one more things because I thought it's interesting and it can be used to asking different biological question. And I'm going just because this is my last two slides, so I will be quick in finishing in 2-3 minutes. So I talk about oncolytic viruses, VSV, vaccinia, HSV, and some of them are currently circulating in human population. So one of them is ORF virus, and this ORF virus is a virus that currently we see 5-6 spillover in human population in Canada from farmers, from animals to the farmers. And what is this? This is a close family of vaccinia virus. It's kind of a smallpox. And when we got this virus, we sequenced it, and it was very interesting. When we blasted to NCBI, it was 70% similar to the NCBI, to the closest virus that we can find. And you know that 70%, it means that what we have here, it's very different from what is already reported. So what I'm trying to say is that we have some virus here, and unfortunately, we don't care. And we don't have enough information. What are these virus? What is their host range? What we need to study this virus and characterize them. And I thought it's good for NSERC. We need doing some basic study, and it can be a good topic for NSERC. So in my NSERC area, I just study ORF virus, and my question is that how this virus it works. I'm not going to talk about any application or how this virus it works. And to start with, let me explain a system that you may heard is piggyback. So piggyback is a transposon system, and how it works, it's very simple. You have two ITR and you have gene of interest. Your gene of interest can be luciferase or can be GFP. And then you express transposase, and this gene of interest randomly, it insert in different location in your genome that you have TTA. So this is how this system it works. And we tried for a system. So I didn't try it. It was another postdoc before me tried it for vaccinia virus. And the question was that how we can make a safer virus for oncolytic virus. And at the end, it ended up to a product that is currently tested in clinical trial. And it was the first product of Translational Biologic, if you heard. And when I was reading this one, I said, I don't like this because it has TTA. How often I have TTA to use this system? But I would like to have a system that I can use it every 5 base pair. It's insert more randomly, and maybe it can be a good replacement for CRISPR-Cas9 system because it's a cheaper. So you have a system randomly inserted, and based on the phenotype that you are inserted, you can just go for that phenotype. For example, I'm looking for a virus that it's it causes fusion, or I'm interested, I can go and find that phenotype. So, and it was interesting that there was a system, its name is Sleeping Beauty, transposon. And why they call it Sleeping Beauty? Because it was originally found in salmon, but the gene, it was inactive. So how they found it? They blasted and they found a sequence that they said, probably this one is a transposon based on the structure of the protein and the sequence, but it's dead. So can we revive it? So they revive it, and then it was wow. It was 100 times more active than the piggyback system that I showed you. And another cool thing that it just needs TA. So now I'm not saying that it's very random, but it's much better than what it was the previous generation. And we started to use this system to insert it in different location, and we were able to find many viruses. So what is the first message you are getting from this picture? So you have many insertion of transposon. So those area that you don't have insertion, it could be essential gene of the virus. So those area that you have insertion, it could be non-essential the virus. So it's a good thing to start. And now you can go and based on what you can rescue, you can characterize those and ask some biological question. So this one, it was my last slide, and thank you so much for listening to my talk. So I'm happy to answer any question.
Thank you, Taha. So, questions for Taha? Go on, go ahead.
Okay, so that's very interesting.
Thanks.
A lot of new things. I differ from your piece here. Learning a lot of things.
Yeah, thanks.
So I was thinking about we're looking at the gut system, right? So is this virus when you infect it, whatever you do, there's any resistance happen? You see some like 5 weeks, you can see the suppress, right? And a lot of models seem to go longer and they go back. Do you see any resistance like this?
So I want to make sure I understand the question correctly. So you are talking about resistance to oncolytic virus. So that's a big problem currently in oncolytic virus, that sometimes the oncolytic virus, they show sensitivity and it's working very well. Sometimes it shows a lot of resistance.
Yeah.
And especially this problem, it's been reported for bladder cancer and ovarian cancer. Because ovarian cancer and bladder cancer, to some extent, they are very interferon pathway positive. So, and then the oncolytic virus is not working very well for them. So resistance, yes, is a big problem for oncolytic virus, can happen because of many reasons. One of them is interferon pathway activation. And what people, they are trying to use to make it, those tumor sensitive, they are trying to arm oncolytic virus with some microRNA or some RNA targeting that target interferon pathway to make those cancer again sensitive. So I'm not sure if I answered it's to some.
Yeah.
Go ahead.
Congratulations.
Thank you.
Your success is really well. So I have a general question. I'm not normally the first person, so this question is maybe really dumb. You mentioned about virus infection and cancer clearance of less prevalence. Has anyone looked at any data from COVID patients? What would patients, people who had COVID, was there any correlation with that?
There are some data at the beginning of the pandemic about the correlation with COVID and all of those. But I don't know what is the correlation, but I remember it was something at the beginning. So
I have another question on your big heat map with all those different chemokines that you put in. So did you put all those different chemokines and generated all those constructs?
yes, and it took, so I forgot to say that. So I was lucky to have a lot of nice people around me. So one of the things that we had, it's about 2,000 mice, and it's just each of them is 8 mice, and it's part of the data. So what we have had was a co-op program, and the co-op program over there is 8 months. So we were able to get, so this is not, it's impossible. All of those construct rescuing each of those, it takes a lot of time. One person definitely cannot do it. So you need to have a lot of help. So this experiment, each of the virus, to engineer them, you need to go for several rounds of purification, and then it goes to rolling bottle, then ultra speed. So I would say for this, just this figure, we have about 5 people, just to 5 people for 6-7 months to work on them. So it's very time consuming, and it's not something that I can do it now. So
I'm asking you. So the scientific question is, you have chosen B16 and CT26, which is the colorectal model.
Exactly.
Where was the colorectal? Because this is intratumoral injection.
Both of them, they are intratumoral, and also we for CD26, and there are two more. One of them is LLC, and another one also that we have the data is MC38. So why we chose this? Because I was new when I started this. So I didn't have enough expertise, being honest. So we wanted to start something. I said, let's go with something that the lab has enough experience. So, and then we just chose these two. Not because one of them is cold, one of them is so, because the best rationale is just choose a tumor that is a cold, hot, or something in between, which for breast cancer, we are doing that. So so this was just a preliminary experiment, I would say, that each of them, now it can be a foundation. So another reason that we use B16 and CD26, because I saw random papers around B16 and CD26. So B16 and CD26, they are a primary model to study oncolytic virus. And why people they chose it, I don't know. But when you look at John's paper in the last 10 years, he always tried these two. When you look at Dr. David Bartlett, when you look at Mike Master, Dr. Mossman, all of those, this is something that they tested first. So to make a link between those data and to start something, so I thought start this one. But for the future, we are doing especially hard ones.
Go ahead.
Great talk, Taha. I'm interested to see that it looked like you got some survivors coming out in the final finale of that QMate inducible triple cytokine treatment vaccine virus. So just wondering, like, that's really cool, really interesting. Did you do anything with those mice? Like, did you attempt like a re-challenge?
Yes, we did a re-challenge and we didn't see any tumor growth. And then we thought, should we work on it more? And then now ask different question about the memory status, about epitope. But we thought maybe for finishing the story is good enough. You see survival. Yeah. But we did it in case if the reviewer asked. That's
great.
Yes, we did. And what we saw we didn't see any tumor growth. Yeah. So it was fully response, and probably it's due to the memory. Memories, probably.
That's great.
Thanks, Ryan.
Yeah, I'm just really curious. Have you looked into actual patient samples when you're looking back into like relatively immunocompromised patients?
That's a great point. So all of those toxicity that I showed, that's a perfect question. I really like why I like your question, because you see the papers, they are trying not the mice, the normal mice, and they are not really representing the immunocompromised. You should go for immunocompromised mice because those are more susceptible to virus growth. So if you want to see toxicity, you don't see in wild type mice. You should go for immunocompromised. And we did all of the experiment in immunocompromised mice. And I would say that if you don't use immunocompromised, you don't see toxicity. So that's very important. For toxicity assay, you should use immunocompromised because those, they show the higher load of virus. So then if you want to see the toxicity, you would see in those.
David?
Yeah, you showed the genome of the vaccinia, the 190 kb. How much of that is extensible to maintain replication confidence? So could you arm vaccinia with all three of those cytokine genes and then sort of sequentially activate?
So that's you are talking.
Yeah. How much of that can you
That's very
arm with the process you were describing?
So first, let me explain you another example, and then I'll answer that question. So first, what is of this one is essential. So we start to cutting from vaccinia, and we were able to cut about 30 kb from here, about 30 k from here. So and that one, we called it SKV, not me, it was, I mean the lab, it was SKV, that was about 40, 50 k less compared to the original one. So now this virus, it has replicating faster because it has a smaller genome and it kills faster. But we show that we observe that when we inject it to the mice, it gets cleared very fast. So this virus, it's replicating faster, but at the same time, this gene also is important for changing the host response, antiviral response. So we can remove it and we can change the time of expression, but being honest, this is very complicated. The knowledge is very limited. Which one should be first? And we are not talking about 1, 2 genes. We are talking almost about the whole genome. It has about 200 genes, and this 20 kb is about 40 genes. So what should be the time? What should be? So all of those things, I would say that is very exciting, but at the same time, it's very complicated.
I mean, you began your talk by talking about nature's experiments. I mean, these guys are being experimented on for billions of years now. So tinkering, it's going to be difficult.
Yeah.
Go ahead.
Quick
Point.
question. Is it?
Go ahead, Sam. The jury's already had one question.
Just curious about your injection. And whether you could do that with NBA.
It was E7, and here the experiment was PFU was E7. It was 3 injection, and it was 2 days, 3 times, 2 days in between. So we did 3 injection, and 2 days distance time between them. And what was the next question? The MBA, could we use MBA? Yes, we can use the MBA. The system, the transcription system of MBA and Tiantan is very similar. But the system that I showed here is Tiantan, because Tiantan is replicating, is more aggressive. MBA also is good, but I believe that MBA compared to Tiantan, it's not very replicative, but it has some other good feature that people they like. Like it's more tolerable, but at the same time, MBA, it has 2 big problems. One of them is manufacturing, because when it's growing very slow, so for production, usually you get 10 times lower efficiency in terms of production. This is one of the problems for MBA. The next problem is that you don't see oncolysis enough. So, and in some cancer model, it shows lower efficacy, but also in some other cancer, you see a better response. So this is the reason that I really think that when you are trying one cancer model, you should go all of the virus that you have already in clinical trial, compare them head to head to see which one it's working better in that specific cancer model.
Jerry, do you want to ask a question?
You haven't.
Great presentation. My mind is blown. And this is not my field. Your talk about safety switches. Does your humic system with the 3 cytokines, does it have a safety switch, or you just withdraw the administration of humic?
So QMET protein, it doesn't have safety switch. It's continuously getting expressed, and that's a great point. But
my next question is, I mean, this is not a fair question because you're not in human studies yet. But assuming you're looking for a certain toxicity symptom, let's say fever, and you stop the administration, is that enough time for the patient to recover? Or you've gone too far already? I don't think you can answer that, but is there any?
But I think it's a very valid question, because when people, they already shoot cytokine stem, it's already too late. So you don't need, so I totally agree. And I think that we should have some other measurement before getting to the point that is out of the control. Then, because when it's getting to cytokine storm, it doesn't matter. You take out the virus, you have a cytokine storm, it keep going. So that's very important. But I would say that we need to have other measurements to make sure that we can turn it off before getting to the cytokine storm.
It's a very quick question, and just because you mentioned there is a difference, bladder and ovarian don't respond as well. So you were talking about non-muscle or muscle invasive cancer, because in non-muscle there is success phase 3 trials with oncolytics going on, right? So do you think because bladder tumors, the invasive ones, they have a loss of 92 where the interferon locus is there, right? So there is a loss of.
So what
does that affect? Because you're trying to put oncolytic in pre-existing tumors, right?
Exactly.
And you're trying to activate interferon pathways in immune and cancer.
We are trying to activate the interferon pathway in immune system, not in cancer cells. Is exactly the point, because in immune system, that one is a good response, but in cancer cells, it's a bad response because it prevent the virus for the production, for spreading. And I don't know the mechanism, which mutation is it, but
Exactly, that's what I was getting at. So maybe because in invasive, there is a loss of that region. So there's, that's why it doesn't, whereas in non-muscle, it's working because it's not that common event.
It's not common, and also you don't have that mutation to make the interferon pathway. So this is the reason that is one of them is working, is not the other one is working. I think it makes sense for ovarian.
It's a complete loss of that region.
So, and also you said about ovarian cancer. Ovarian cancer, we are providing the virus with David Huntsman, and he just gave us the data. So what is exactly happening is that because this is the interpretation that he provided, that his data shows a lot of heterogeneity, and some of the cells get infected very well, show the replication of the virus. Some of them, they are very sensitive. So he said that some of them very sensitive, some of them very resistant. So based on what the data we got from him, it's very heterogeneous tumor microenvironment, cancer cells. And what is the reason? I don't know exactly what's the story behind.
Yeah.
Bye-bye.
Amazing thought. Thank you.
Yes, thank you.
With your system of turning it on and removing to turn it off, with 3 cytokines, you just see that it'll go up, the cytokines will go up, or they'll come down. Is there a way that you thought of to be able to regulate how much one will go up versus another?
That's a perfect question. So this is the next question. I would say that you get to the point that you think that, okay, now I have enough novel data to finish the project and then expand or have multiple, because there are 2 things. So you have cytokines, you have a table for breast cancer, hot, cold, or something in between. And now you want to say that which one should be combined and which one should be on, off, the timing, the level, and all of those games that you can do. I think you can do it forever, but you want to do to some extent to show the field, to convince the field of oncolytic virus that the time, level, and combination of cytokine chemokine is important. Now what they do, they have a virus, they have a cytokine, it has an intelligent tool, is expressing all the time. So this is not the way. You need to separate everything and find where, when, how much. And this when, where, how much, I think it was missed. And now we have this tool, we can work on it. And yes, yes, this is a plan, and the time, the level, and combination. So the cocktail therapy of the 3 also. So because you said that these 3, each of them, but also it can be combined with other virus. So
that entire table that you have, are you gonna?
Not all of them, I think then but to some extent you can choose.
Right.
For example, you saw that some of those cytokine on the bottom, it increased the efficiency. So, and those chemokine, they are recruiting immune system. So now it makes sense. First recruit the immune system, then activate them. It doesn't make sense to do it vice versa, right?
Right.
So now makes sense. Recruit them. So for recruiting, you do a screen. For activation, you do the screen, and then you go for this one, and you have a time, first, second.
So he needs another 50 or 60 people to do all the 35, 40.
And I don't know if I have.
I think I'm gonna cut off questions. People, pressing questions can come up and ask Taha afterwards. And but I'd like to thank Taha for an amazing talk. And as we were discussing at lunch, this idea of people got to learn to get along